डीएनए जेल इलेक्ट्रोफोरेसिस और उसके बाद स्टेनिंग के करने के बाद जो लास्ट स्टेप है इलेक्ट्रोफोरेसिस में, टू टेक द पिक्चर और फोटोग्राफ ऑफ दैट पर्टिकुलर जेल विद स्टेन्ड बैंड्स ऑफ द डीएनए, ताकि हम उसको परमानेंट रिकॉर्ड में रख सकें। जैसा कि आप जानते हैं कि यूवी लाइट का जिक्र बारहा आया है कि यूवी लाइट को यूज़ किया जाता है टू डिटेक्ट द बैंड्स ऑफ द डीएनए, वेदर इट इज स्टेन्ड विद एथिडियम ब्रोमाइड और इट इज स्टेन्ड विद साइबर गोल्ड। After this detection, photography is required. So we are going to talk about how it can be done. UV light के हवाले से जब बात की जाती है, UV light can be used in two different ways. One is the transmitted UV light. The second one is the incident UV light. In both the cases, the wavelength which is used is 302 nanometer, which is available in most of the commercially supplied these transilluminators. If you look at this picture, this arrangement shows the transmitted UV light which is used for taking the photograph of any gel stained with the ethidium bromide or with the cyber gold. This particular box is basically the space or is basically the stage of the UV lamp. This is the source of UV light, the tube which emits the UV light. Then at this position we have a transparent shield through which UV light coming from the base can pass, and this is the gel which is lying over the transparent area. So the UV light coming from the source passes through the gel, and when it passes through the gel, it illuminates all the DNA bands or RNA bands in the gel, and these are visible as orange bands or orange to red spectrum bands. Then there is nowadays available the digital cameras which can picture your gel, and that picture contains all those fluorescently labeled bands. The second method or the second arrangement of the structures or of the components involved in incident UV light uses the UV lamps not below the gel, rather these are above the gel. Through these lamps from both the sides, the UV light is emitted and is going to hit the gel, and then the bands are fluorescent here, which is recorded by this camera. In this case, no lamp is found in the base. There is a spot which is kind of black in color, and then above that spot there is lying the gel, and above the gel on the lateral side there are the source of UV light. This is the system for incident UV light. These were the conventional methods which were used for photography or for detection of the DNA bands in the gel. Nowadays, very sophisticated equipments are in laboratory, like they are referred as gel documentation system. They are fixed or installed with the cameras, and they have a dark chamber where you can place the gel, and after emitting the UV through the transmission system, the camera is going to detect the picture or is going to detect the DNA bands, which are later on printed by using thermal printers. One step ahead, the gel documentation system, they are connected or they are supplied with the computers. These computers have the installed softwares, and this kind of system has made it possible to observe the DNA bands in real time and to manipulate the screen, and then to take the picture in different formats, save it in different formats, and even then we can manipulate the image using different softwares. We can label them, we can analyze their density, and number of other functions can be performed. The detection limit can be improved with this improved sensitivity, as if you prefer cyber gold stain over ethidium bromide stain, then it can give you an improved results. But again, यहाँ पे वही बात आती है cost effectiveness की. Use of ethidium bromide is cheaper as compared to cyber gold. We can compare it like 5 cents versus $100. ये वजह है कि साइबर गोल्ड की बजाय हमारे पास एथिडियम ब्रोमाइड स्टेन हर वक्त लैब में मौजूद होता है और एक्सेस में मौजूद होता है ताकि हम उसे डीएनए जेल्स की स्टेनिंग के लिए यूज़ कर सकें.